glycoshape, a resource to restore or rebuild like protein glycosylation on the 3D structure of glycoproteins. And I'm going to try to explain to you why is that important. And first of all, like I'm sure I don't have to tell most of you how magnificent, mind-boggling, and complex is the human. I'm just like restricting myself as human cellular glycome with all the many different types of glycans structures, functionalize proteins and allow them to perform optimally their function and adopt their preferred structures. So how glycans, like in their inherent diversity of sequence and structure and branching and their like inherent complexity performs these functions is still a mystery. And I like to use this slide. I've seen somebody at the SFG using it, but I'm using it longer, so I'm gonna keep on using it. And it's just like a slide about how like the glycans and the glycome is still considered or can be considered as a dark matter of the biological universe, which is not my own sentence, but it was written in Essential Glycobiology by Ajit Varki and Stuart Kornfeld. It doesn't get much better or more clear than this definition to give us an idea of how mysterious is still the glycobiology is still the field of glycobiology. So why is that, or why in some part that is that glycans, or actually most glycans, are intrinsically disordered from the structural point of view. And the most come from an edit that I got from Mark von Idstein that knows quite well about one thing or two about glycan structures. So glycans are intrinsically disordered because they're very flexible. They are inherently flexible. So it's like a bit like this photo that was taken on a busy street in New York with a 5-second exposure. And people walking down the street are actually blurred, like because they're walking too fast relative to the shutter speed. So you cannot see them. You just see the blur. But you can see the people that are actually stationary. And this is a bit the same for glycans, as they can be partially resolved by crystallography when they're structurally restrained. So for example, where they are bound, and usually that is limited to a couple of monosaccharide, like 2 or 3 at best, usually. Capturing more than that is quite a remarkable feat. And in terms of cryo-EM, things are actually extra complicated because of the kind of structural procedure and the inherent like protocol that is required to resolve the structure by cryo-EM. You have an additional problem adding to the disorder, which is the macroheterogeneity of the glycan. Different glycans are often in the same glycosylation site as that's a feature of glycoconjugates. So why do we think it's important to get the 3D structure? And why do we work so hard, especially using very expensive MD simulation to resolve what it cannot be resolved experimentally? Is because we think that the 3-dimensional structure of glycans is actually an inherent part of the glycocode. So like the way glycans facilitate particular biological function through their 3D structure and dynamics. And that's how, for example, they're recognized and they establish interactions, specific interactions with lectins, with antibodies, with carbohydrate active enzymes or cases. How do they perform folding or facilitate folding and affect the structure and the dynamics of proteins? And how are the first port of entry for bacteria, toxins, and viruses infection? And in that case, actually, was as all of us pretty much following the new virus scare that is rising up now, that it was actually flagged in Blue Sky like recently, like I think it was yesterday, and or 2 days ago, about the avian influenza H5N1 that has acquired a new mutation
that might allow binding to actually human epitopes. And this is like, it doesn't offer me, this offered me the opportunity to show you how structure matters. So the epitopes have the same sequence in terms of what monosaccharides are bound, but you can tell that a 2-3 like linkage is a completely different 3D structure than a 2-6 linkage that is prevalently bent. Meanwhile, the avian linkage is prevalently straight up. So a mutation would allow to accommodate different epitopes because the glycans have different structures, not different sequences. So in order for you to do the same without having to learn what is molecular dynamics and having to, which I invite everybody to do that. It is a fabulous field of research, but again, it's a technique that it might seem easy, but is not as easy as it seems to use, especially is extremely expensive computationally. We created a resource, it's called Glycoshape. So Glycoshape is a glycan structural database and a toolbox. So we have we provide the structure, the 3D structure of the glycans derived from extensive molecular dynamics. And in this at this date, like I checked yesterday, so like our website, we have live 623 unique glycans structures. So most of those are from the Human Glycome. So at this point, you can easily rebuild any type of human glycoprotein or a protein of, for example, virus that affects humans. And but we have at least one example from Alpha. So like this is maybe something that like we might have the epitopes that you're looking for, or the glycan you're looking for, but if we don't, please contact us and we'll build it for you. So you can use these structures on their own, like for example, for docking or for just looking at them, like so for a structural alignment, whichever topic you want to, or structural analysis you want to perform. Or you can use those to rebuild a glycoprotein with a tool that we developed. Is called Reglyco, that I'm going to talk about today. So you can source by using to use Reglyco, you can use a PDB structure, just like fetching it through the PDB ID, or an AlphaFold structure, fetching it through a UniProt ID, or the protein of your choice if you're a structural biologist and you have your own structure, or if you modified your structure, another structure as you wished to do. So this is mostly the work of all my lab, like everybody, including myself, make glycans all the time. But it's the design of the old tool and the database and the algorithm is largely the work of Callum and Ojas that have invested all their postdoc and PhD respectively to do this. Like recently, Callum left us for his first academic position, so we're proud of him, and Oja's taken the reign of everything and is running it like a pro. So to give you an idea of how spread out is the use of Glycoshape, is we have like lots of people working with reglycosylated proteins around the world. Like, so this is a map that describes the usage of Glycoshape so far. So this is up to October 2024. We are almost 1 year old because we launched in December 13, 2023, and since then we had quite a bit of humbling success. So like people are using it and we're delighted about it. And on average, like the usage before the paper was like Impress, like the Nature Methods paper that describes Glycoshape was Impress. We had an average of 10.3 visits per day. So now we have a bit more, so we hope to keep up the rhythm. So, and I like to notice, like, because I like maps, that we can cover north to south from Umeå to Melbourne. So that's pretty wide, like, and so we're excited about this.
So to give you like an idea of how the database work and how you can use molecular dynamics, extensive molecular dynamics to create reliable structures, I'm going to give you the rundown of how this glycan analysis pipeline that Ojas and Callum designed works. So we have adopted a multi-microsecond cumulative sampling protocol, a bit inspired by grid search, where we independently analyze all the degrees of freedom, all the dominant degrees of freedom that describe the dynamic of glycans. So, and the sampling is uncorrelated. So we like every structure of the glycans, every glycan structure from disaccharide on is simulated for at least 1.5 microseconds. So it's extremely extensive sampling. So we put all this data into a cumulative trajectory, into a graph matrix, so a distance matrix, and we flatten the lower half of this matrix into an array because of the dimensionality reduction required. This is not quite enough, so we use PCA, principal component analysis, to reduce to 3-dimension. And we use clustering. So we cluster these PCAs, 3-dimensional PCAs, into using a Gaussian mixture model that you can see around here, like leads to clusters. So for each one of the 600 and odd structures that you have multiple conformers that are populated within different percentages. So you have a dominant structure or dominant shape, which would be for example, the bent one of the cellular lactose that is recognized by H5N1, or like, this straight one that is recognized for the 2,3 alpha cellular lactose. So these structures are all available to you, so from the lowest populated to the highest populated, well, the other way around. And there's extra pages on the relative to each glycan that is identified primarily by its glycan ID, but also by other identifiers like UPAC or glycan names and so on. They're available to you, sourced by different glycobioinformatics resources on the page for each glycan. So you can get on Glycoshape through this link, and we don't even get you don't have to sign up, you don't have to do anything, you have to just use it. So how does this work? So now we have all these glycans and all these glycan shapes are like the dominant, or the summarized kind of like the dominant structures for each particular glycans. So Reglyco stores this database to rebuild protein glycosylation. So, and I'm just going to show you here how this works in the most unusual, but probably the most or the luckiest scenario. So where you have the protein structure, and that's commonly now with AlphaFold is a common thing to have, the protein in the good or the bad. You also have a list of the glycosylation sites, and this is actually not that common because you need to have glycoproteomics data to accompany your structural biology data. And better than that, what glycan goes where. So what type of glycan is occupying each glycosylation site. So with this information, like that, if you're blessed with so much information, you can go to Reglyco and fetch the PDB ID, of your protein, or the UniProt ID of, if you want, the AlphaFold or your own protein, and start rebuilding or connecting the glycan of choice. So you select the glycan that you want. So glycans are linked to the protein with by genetic algorithm that minimizes a loss function. So it's a repulsion potential, but it works so very well, not only because the structures of the MD structures are quite accurate, but actually because the linkages are very good. So the linkages are derived from a database, from a crystallographic database, or produced separately by MD for each type of glycan. So, and the crystallographic data from Privateer. So And in this case, like, so the repulsion potential would exclude, for example, the most populated cluster, in this case, corresponding to this very busy tetrantennary hypersialylated and glycan, but the second highest populated cluster would fit. So it's the one that is going to be put in to your protein. And the O glycan would be the same, will follow the same procedure. So the thing is just like if you have like the glycosylation profile already available in UniProt, there's a little button would appear that allows you to do everything, all the glycosylation site at once by following the UniProt information or annotation in one click. So that's quite handy, especially if you're in a rush. So this will be like, will take seconds, obviously, depending on the complexity of the glycans that you choose, the complexity of the structure, how densely glycosylated it is. But usually it will take like a couple of seconds if you have like sparse glycosylation. So this is the most common case. So you got the protein structure, but you have no idea where is glycosylated, and you don't know what glycans goes where. So this is all of us pretty much every day. So in this case, you are brave, you choose your protein again, and you want to see, like, or test what end glycosylation is possible. So we cannot really help you for all glycosylation because that doesn't follow up any sequon, but we can definitely help you with end glycosylation with a tool called GlucNAc scanning that uses the power of Reglyco to scan the surface, the protein surface, and to see if one glucac, which is the root of the glycan, of an N glycan, fits. So in this case, for example, you see that in this protein, the 47A sequon can be occupied because the glucac can be housed like quite readily. Meanwhile, the 90A, it cannot be occupied because it's actually here and is in the interior of the protein or the inside the helix packing. So it doesn't fit and is not occupied, which is actually the reality of things. So at this point, you choose your N glycan of choice, this A2 glycan, like very easy peasy, and the NO glycan that you want because you know that this position is occupied, and you put them back. So this is what I just said, sorry. And so GlucNAc scanning actually works extremely well. It scores 93% against experimentally profiled structures in the PDB for many different reasons, fundamentally because glycosylation actually is involved in the folding of the protein, and it only occurs where it's accessible. So I'm going to show you how this works in a practical example. And this is an example that's also discussed in the paper. And it's the example where we had the most, headache, in the sense that usually this works like a charm. Everything is consistent with the experimental data, except for CD16B, which is the gift that keeps on giving. It's a protein that is a lot of information on. However, it's quite difficult. But this example actually illustrates how Reglyco works so well, and especially can provide also information about the inherent quality of the structure that you're working on. So we decided to work on a PDB 1E4J that was resolved at 2.5 angstrom resolution by crystallography. You can see it here, like in this picture. So we know the glycosylation sites, and we know actually what glycosylation sites are occupied and what glycan goes where from a very thorough analysis in Manfred Wurer's labs. And this analysis, mind you, I like to underline, that was actually done on neutrophil-bound CD16B that was released upon glycoproteomics analysis. So it corresponds to the neutrophil-bound CD16B. So when we run the Reglyco on 1E4J, though, we get 2 false positive and 2 false negative. They're actually quite indicative of the difference between what it means working with a membrane-bound structure and with a recombinant protein. So if we start with the false positive, actually, we see that these two sites are actually quite accessible. So Reglyco is going to put easily a GLUCNAC in these positions. But and if we if we scour the literature, we see that if 16B is actually resolved recombinantly, it's glycosylation, like these two positions are occupied in the recombinant 16B. Therefore, it's actually a point that the actual environment where the CD16B is active, like the membrane, like so N35 is extremely proximal to the membrane, so it could hardly be glycosylated when this protein is bound to the membrane. And N64 is more difficult to explain in terms of proximity to the membrane, but again, the dynamic of the system might prevent its glycosylation because, again, closeness to the membrane. So, but if we looked at the false negatives, actually, these are informative of the structure, the PDB structure, or let's say the assignment of the atoms in a PDB structure. So in the case of N45, the fact that I cannot put a glycan here is not due to any structural difference other than the wrong assignment or the like the non-glycosylation friendly assignment of N epsilon on Ne2 or OD1 in the asparagine side chain. So if we looked at another structure that is like this PDB6EAQ, which was solved with a glycan, like with a glycan root, like on this asparagine, and then 45, we'll see that actually the N and O are inverted. So we provide like on our website like a swap tool that allows you to swap these coordinates in one shot and reload the PDB. So to try if you can put a glycan at this position, or the non-fitting is due to the wrong assignment of N and O positions or actually coordinates. So one thing is just like, as every crystallographer knows, the assignment of these two atoms is completely or virtually arbitrary, because you cannot really tell which one is which from the density. So in this case, in the case of this part here, like so the wrong assignment or the let's say false negative N74, the problem is actually that this structure, like, I mean, this area is very, very poorly resolved. So, I mean, like in this case, like the density was fitted with a structure, but in the case of the pink structure, for example, this density was not fitted because it was very loose. Like, so you can feel entitled to rotate this asparagine, which is actually wrongly packed against another loop anyway. It's very odd. And in this case, if you rotate this, if you perform a rotamer search, like the glycan will fit like a glove. So the thing is just we didn't automate it like the rotameric search because we don't want to receive like a million calls from crystallographers seeing people ruin my crystal structure. Like, so structures can be elaborated on only if you are knowledgeable about it and you are very well advised or very well advised. So we cannot really automate it on our website. That's the decision that we made. So based on this, like, you can rebuild your CD16B as it is in neutrophils bound molecule with the glycomics data indicating what type of glycan goes where, which is actually makes it so much more beautiful than it was before. So in case of very complex systems, so nothing is more complex that HIV, one envelope, like so, Reglyco works very well as well. And this is work done by Joel and Max. So I got this slide from them, and it's published now like on
bioarchive. So you have like that they are careful glycoproteomics profile of the M329 SOCIP has guided Reglyco to rebuild this fully glycosylated protein and that they were studying. So what about AlphaFold? So like in the actually like realm of AlphaFold, we have that most structures or a lot of structure are actually like just like modeled with AlphaFold. So how does Reglyco or how does an AlphaFold model allows for reglycosylation? Well, we argued this point like a couple of years ago with in collaboration with John Aguirre. And what we figured out is that because AlphaFold was trained on structures that carry, like a lot of them carry cofactors, carry glycans, carry all kinds of PTMs in the PDB, AlphaFold should implicitly know about how to build a protein that carries glycosylation without knowing explicitly about glycosylation. So in theory, for a high PLDDT score, AlphaFold should be glycosylation ready. So we eviscerated this topic in our ultimate testing that is presented also in the paper, where we analyze 3,415 proteins, like 12 over like almost 13,000 N-glycosylation sites. And we saw that indeed, in 85% of the cases, the clashes are easily solvable by Reglyco alone, which is able to put any glycans on the appropriate glycosylation sites, so predictive glycosylation sites on AlphaFold structure. So this is an exostosin-like 3, we chose this example as an example that because it has crystal structure, cryo-EM structure, AlphaFold structure, and in case, for example, in this case, the AlphaFold structure didn't have a good orientation of the asparagine, which prevented glycosylation, a known glycosylation site. So in this case of the AlphaFold, you don't even have to do a rotameric search. Now you can use AlphaFold 3 that gives you 5 structure. One of them has for sure like an optimal structure or a rotated version of this torsion of this side chain, but you can also use AlphaFold as we did this test before AlphaFold 3 was issued. And yeah, so fundamentally, why restoring 3D structural proteins is important, because like from the static point of view, just looking at a very well, crafted 3D structure of a glycoprotein gives you an accurate description of what the glycans might be doing, and to understand or give you very good hints of understanding structure to function relationships. Also, having all these multiple glycan structures in a database can be useful to understand binding specificity. For example, going back to the H5N1, see how different mutations might change, or test how different mutations might change the specificity of the hemagglutinin for binding different epitopes, or any docking that you want to do with those structures that are actually the populated structure in solution. But what I want to talk to you about is something I haven't talked in, actually, believe it or not, ages. And I want to go back because I started with viruses, and I want to, let's say, finish with the virus that possibly I know best, which is the SARS-CoV-2, in particular, spike protein, and see how the glycans control the dynamics and the binding of this protein, which is quite a unique feat. So like another example of the binding, I'm just putting it here because it's been recently posted on bioRxiv, it's just how glycans bind proteins, and it's not a protein-protein binding, but it's actually a glycan that binds the protein. In the case of CD52 and HMGB1 box B, where actually the hypersialylated N-glycan and O-glycan favor the binding or the formation of the complex between CD52, HMGB, 1 box B. This is work with Nikki Packer and Len Harrison, if you're interested. But again,
going back to the viruses, this is like what I'm going to talk to you about today is actually how glycans affect the structure of a protein. Removing glycans disrupts the structure and the binding of a protein. And actually including the glycans into your viral analysis of the viral mutations and evolution can help understand or epistatic control, which is like something that is always difficult or impossible to understand if you don't have a full picture of your protein that includes glycans. So we'll see that you're probably familiar with this picture in particular because it's an iconic picture of the. I can totally say it because it was in the cover on the cover of Essential for Glycobiology. So iconic picture of the spike protein by the ultra talented Lorenzo Casalino. So they're depicted in blue just to stand out how the glycan shield the spike protein that is in cyan, and you can see like the shaved version is quite miserable and it wouldn't even exist if it wasn't for the glycan that helped it fold. But what we figured out actually in that paper is it goes beyond shielding. So it's something that the glycan do, and it's the first case where a glycan actually was, or a set of glycans rather, that were highlighted to affect the function and mechanics of this spike protein. In particular, this was originated, or the curiosity was originated by the glycoproteomics profile that Max Crispin published in early March, like 2020, bioRxiv, where I saw this incredibly weird man9 in this position at the top of the spike, and I thought it was like quite unusual to have such an enormous oligomannose in the wrong spot, or what I thought it was the wrong spot. So that's prompted us to rebuild the spike in many versions. And what we saw is that this glycan that I'm depicting in green, following the SFG nomenclature, because it's actually like a oligomannose, like, you know, forgive my glucnax, like, so everything is in green, actually crawls inside a pocket that is formed by the opening of the RBD, or receptor binding domain. And so in this case, in this video that I should change colors of, like I represented actually this man9 in pink. So, and filling that gap supports the RBD in an open position, which facilitates ultimately ACE2 binding and infection. So this insane amount of MD simulation and heroic effort was actually cited by biolayer interferometry data from Jason McClellan's lab we partnered with, which who did express like different mutants to highlight the function of different glycans. And this obtained that by removing that octopus glycan that crawls inside that cave with a single mutation, you would triggers lowering of the binding affinity to ACE2 down to 60% relative to, at this point, it was the 2P S spike protein. So a massive dramatic change of affinity due to the fact that the spike just doesn't open. I can tell you that for sure, that that is actually caused by the absence of one single glycan in 22 glycans per monomer. So that's pretty much nails it, that glycosylation is fundamental, not only for shielding, but also for function. So this triggered in my head like an absolute curiosity about how glycans or the shield evolves in a system that is where the shield is not just shielding, so it doesn't promote evasion, but also promotes fitness. So like it just makes this protein functional. So you can see if you go to many mutation up to GN1, and the mutation that are circulating now do not differ from that. You only have very rarely changes in glycosylation that actually affect dramatically either the evasion or the fitness or both. So the first mutation I actually highlighted the Wuhan because Wuhan is like can be considered as a mutant relative to previous or other subacroviruses where it comes from. Shows that glycans that are mutated are actually dispensable. Glycans that are not used for function, so they're easily dispensable. So those can move. So like in this particular case, Wuhan has lost one critical glycan that was not critical for its function at all. Actually, losing it improved opening of the RBD, which became energetically favorable. So, and actually, that is like removing that glycan from a subacrovirus that has it, like the one in bats. The spike from bats, like just like from the virus that affects bats. I don't remember the name now. Like increases infectivity by about 190-fold. So we looked at another highly conserved glycan that also Romy Amaro wrote like fabulous work with in collaboration with Lillian Chong and Jason McClellan again, which is highly conserved in N343. So in this glycan is like they found through extensive simulations that gates the opening of an adjacent RBD group, so helps it open and close, favoring like the binding to ACE2. So removing that glycan actually reduces dramatically again the like the binding to ACE2 relative to the Exapro in this case, the Exapro, which is like the most stable spike that you can find, also designed by Jason McClellan. So what we wanted to see is actually what does this glycan do to its own RBD, not to the adjacent RBD? Does it affect the structure of the RBD? So this seems like a quick and easy question, but it was like it took almost 4 years of work between my group and John Klassen's group in the University of Alberta, because the RBD actually is an extremely difficult, structurally difficult system, as it combines a structure domain or structure part of it to a disorder, a highly complex disorder domain. And you can see here in this kind of like more see-through cartoonish representation. The structure domain hosts the glycan that we are working on, which is like 343. And this glycan has a lot of contacts. So it just like, it binds over on top of the RBD over this region here, and like this is the asparagine that is occupied, and these are the anchoring point, in particular these serines that are anchoring point for the glycan that just like covers like a lid. So you have that in the bound conformation. So now when it binds ACE2, this disordered motif is actually quite folded, the RBD has like a system of residues that favor this conformation upon binding, which is an hydrophilic patch and hydrophobic patch. The hydrophilic patch is actually the most intense network of salt bridges I've ever seen, that you can see here, all this charged residues that interlock these loops in a pretty much very stable contacts. And the hydrophobic patch is actually produced by the stacking of these residues here that actually also extremely highly conserved, to the point that you might have heard about like some flip, like aptly named flip, like a variant where they are not changed. They're actually like they're swapped, like these two residues are swapped. So they're swapped, but they are not never changed. So the disruption, this all indicates the disruption of this fold compromise binding and infection. So like if you disrupt this RBM part or the RBM fold, you will lose binding to ACE2. So we did try to understand how that works and if that depends on the presence of this N343. And we removed the 343 from Wuhan, alpha and beta, and ran lots of molecular dynamics. And what we saw is that because the glycan actually covers a very greasy or hydrophobic part of the RBD, and he actually protects it, forming actually an interface that through the fact, like based on the fact that glycans are actually, or carbohydrates are amphipathic. So they can bind the underlying, doesn't mind the underlying hydrophobic side, but it actually is able to bind also water. So if in the absence of this amphipathic kind of bridge, you will have that the structure of the RBD response trying to push or like prevent that, to close this access to this hydrophobic core, or this hydrophobic belly as we call it, and pushing these two helices together to prevent water access as much as possible. But this actually connects a connected effect that makes this or affects directly the RBM folding, and it actually misfolds it. So we decided to test this MD evidence through binding of this GM1 and GM2 that we know from work in John Klassen's lab and collaborators, binds in particular this site that we identified through MD, so which is this site here. So upon misfolding, this the RBD would lose binding to GM1 and GM2. So we indeed see this. If you remove the glycan, binding to GM1 and GM2 is lost completely. So that supports the evidence that we found. We tried on other mutation, other variants that were coming up all the time. So at that point became a race against the viral evolution. We saw in Delta that immediately actually the glycan became quite unhinged. So it started, instead of being hooked on the belly, like so just protecting it, like and being quite stationary, it became very mobile, like, you know, covering almost the whole surface of the RBD. And this is, it seemed that it was a bit like, or less hinged or less stable. So it's not as anchored as I wrote there. So Delta has one mutation that is quite interesting, which is this mutation here with L452R, which actually changes the junction between the disordered RBM and the structured motif. And it kind of like stabilizes the connector between that helix that pushes in the absence of the glycan, and it triggers like the disorder here. So it makes this junction way more stable. So removal of that, like totally expected, the removal of the N3 from Delta actually doesn't cause any effect in the GM1 binding, which is not affected at all. It binds the same presence of glycan or not. So Delta N343 becomes dispensable, like because it's not indispensable in order to bind ACE2. They can be lost. So Omicron doesn't bind, like to conclude, Omicron doesn't bind because of GM1, GM2, because of another mutation that anchors GM1, GM2 in place. But Omicron has a lot of mutations, and actually all the residues that are actually anchoring, or that we saw anchoring the glycan in Wuhan, are gone. So they're here mutated, like you can see here in this pocket. All those anchoring glycan residues are gone. So what we get is that actually, because of how, like, you know, of the different structure of Omicron in, like, of the Omicron RBD in general, like, and the fact that actually, like, all those anchoring S are actually swapped for hydrophobic residues, so they're able to pack much better within the hydrophobic belly of the RBD. There's no difference at all if you have the glycan or not. The structure is very sturdy and it remains the same, as well as the fold of the RBM. So we have no structural effects upon loss of the 343.
In addition, like the newest variant and the variants that are currently circulating, so that you know, they have two glycans that actually work, another glycan that actually works in combination with the 343. So, and that actually it's actually positioned below the 343, and it covers part of the epitope, or part of the RBD that wasn't covered before. So it makes it lose, like, it just loses recognition by neutralizing antibodies. So in Omicron, N343 again is dispensable, and the presence of this extra glycan might actually favor its disappearance or its mutation, which is or its movement along the same backbone position. So new variants on the horizon that might have different glycosylation sites. So to summarize it all, I'm sure I don't have to convince you why you should rebuild a whole glycoproteins with the glycans and all, and I hope you can use, you'll be able to use the Glycoshape to do that. This is a slide that I always use, but the credits goes to Ben Schultz, the University of Queensland, for the genius of this slide. And I'd like to thank you all for your attention, all my students and collaborators on this project. Thank you very much.